Hello and welcome to this lesson, which is actually the second video lesson on the distribution of organisms. If it's part 2, you must have seen part 1, so you can continue to part 2. Otherwise you need to pause this and go back to part 1 of this video. Okay, so if you've gone through this video, you should be able to state what a transect is at the end of the video, hopefully, and you should be able to analyze data and explain and describe the distribution of organisms. Then you should also be able to explain the distribution of organisms, giving a list of possible reasons why the organisms are spread in a particular way in a habitat. And you'll also be looking at methods used to estimate the population of organisms and be able to say what's good about it, what's bad about it. That's evaluation. Before we go any further in the estimation of animals or organisms, we need to know about sampling tools which are used to estimate population. You might have seen this in school. It's just a plastic frame which is usually 1 meter square of area. This is laid randomly in field or in a place where you want to sample the organisms, and then you just simply count the number of organisms inside. And if you count the number of organisms inside and take a few more samples, you get the average number of organisms per square meter, and then you multiply that average by the total area of the field. This is covered in part 1 of distribution of organisms. So if you're not sure, please go back to that video and see how exactly a quadrat can be used. Now you also have another sampling tool which is called a line transect. Now a transect is usually described as a straight line. It is nothing but a straight line of a measuring tape, or it can even be just a string but which has got regular markings on it, evenly marked. A transect such as this is normally used to find out how an organism's population or distribution varies along the line in a habitat. So if you go here, if it is more, less, that kind of distribution can be measured along that straight line. It gives you a guidance as to across the habitat that you're interested in. Okay, let's take a close look at how a transect looks like. As you can see, you can place quadrats along the length at regular intervals. You can see the 1 meter square quadrat, and then you can count the organisms inside, possibly snails or things which don't move very fast. You can also sample, for example, a plant species, particular species or a variety of plant, and see how its distribution varies along the length of this habitat. So it gives a kind of proper guidance and regular intervals. It's like markings where you can go along the length at regular intervals and count the number of that particular species that you're interested in each of this quadrat. And then if you look at the data, it may tell you a story. For instance, you may find that that particular species live more in a particular area, for example here, and maybe even more here, and then becomes less and less here. And then you look at possible explanations for that kind of distribution. That is how a transect can be used. So a transect is a straight line across a habitat that has regular markings on it. Say every 5 meters you will have a marking on, so you can actually sample for a particular species of an organism along the markings. A quadrat is just a square frame used to count organisms inside the area of that frame, which is a quadrat. Okay, so normally many quadrats are used at once. As the sample size is bigger, you get a more accurate estimate of the population. Very often in this topic you will be tested on these statistical terms: mean, median, mode, range. Now mean is the same as average. So let's have a look at an example to understand these terms. Actually, two examples. Let's have a look at a first set of sample: 2, 2, 3, 5, 5, 7, 8. They are just numbers, but you may want to consider them as number of living things. For example, number of tortoises, or anything that you want to imagine them as, because we are looking into distribution of living things. So now, given that set of numbers, and this is what your sample is, what will be the mean of this? To find the mean, all you have to do is add them all together and put the total. So the total in this case is 32. So there are 32 organisms in your sample. Divide by 7. Now, why do we divide by 7? Because you have 1, 2, 3, 4, 5, 6, 7. 7 samples here. Okay, so you divide by 7, you get your mean or average, which in this case is 4.57. And if you want to round up to the first decimal place, you would have to write that as 4.6. Okay, so that's your average number of organism in your sample. Next, median. So this is your mean. We've done the mean, done that, and now we're going to be looking at median for the same sample. Now, median, to find the median, you need to put the numbers in order, in the increasing order. It's already in that order, as you can see here. 2, 2, 3. If not, you have to put this in increasing order. Okay, 4, 7, 8. Now I have just repeated this because it's already in order. And once you've done that, you look for the middle term. So if you count 1, 2, 3, 4, 5, 6, 7. So you have 3 here and 3 here. Your middle number is that one there. Okay, so that is the median value. So for this set of sample, that is the median. Okay, mode just means mostly occurring value. So to find the mode, you have to look for which one is the most reoccurring number. So in this case, it's 2 and 5. So you got 2 modes here. They appear more than any other number. So in this set of sample, you have 2 modes. Sometimes you get only 1 mode. Very often you get only 1. Finally, what is the range of this data? Range is the difference between the smallest number and the biggest number. So in this case, 8 take away 2, that gives you 6. So the range equals 6. So we know all these terms for this set of sample. Let's have a look at another one very quickly. Here is another set to familiarize ourselves with these terms: mean, median, mode, and range. Okay, so we have that set of samples. The mean would be, just add them together and you have 25. And then if you divide that by the number of samples, so in this case we have 1, 2, 3, 4, 5, 6. You have 6 samples. So you divide that by 6. Your mean would be then 4.166 or 4.12. So if you round up to 2 decimal place, that would be the mean number of organism in your sample. Okay, so the next thing is mode, as we have done before. Median. So we've done mean. Next we are looking at median. Okay, so median is, as you know, the middle number. So in this case, if you put that in the increasing order, 3, 3, 2, 3, 3, 4, 6, 7, you have there is no middle number. So you can see 3 here and 3 here, so there's nothing in the middle of this. So in this case, you have these 2 as a median number, so 3 and 4. But you can't give it as it is as a median number. You add these 2 and divide by 2. So in this case, 3 plus 4 equals 7, divide by 2, and that gives you 3.5. So that is the median. Okay, so median in this case, you have to add the middle 2 numbers, as there is no one single number. as the median, which is in the middle of everything. Okay, the other thing is that we need to know is a mode. Okay, again, mode is the most often repeated number. So in this case it's easy. As you can see, this number here, 3, occurs twice, and there's no other number which appears twice. So the mode in this case equals 3. So this is median, and median calculation is shown here. You look for the middle one. When you want to calculate mode, then you look for the most occurring number or sample, and then you have finally the range. So the range in this case is the smallest number take away the biggest number. Bigger number take away the smaller number. So in this case 7 take away 2 is 5. Okay, so that's your range. In this example, let's have a look at a practical problem, the type of a typical problem that you are presented in an exam. You know, badgers live in woods in the countryside, and they live typically in holes and burrows like this, and that's an example of a badger house, and they're known as set. So that's SETTE. So in this example, scientists actually trying to estimate badger population in 4 different types of wood. Okay, 4 different types of habitat. And let's have a look at and understand what distribution actually means. This real map of badgers in some place in the UK with them, it gives an idea about what distribution means. Here you see a big, huge area where badgers do live, but you don't see them evenly distributed. You can see clusters of them. You know, each of these markers on the map represent a badger set, which means they prefer to live in certain areas, normally dictated by a few factors that determines where they are found. Normally for animals, it is food and the possibility of predators. So if there are more food available, you'll find more of them in a particular area. If there are more predators available, you'll find less of them in that area. So let's have a look at the factors that affect the distribution of any living thing. These are the factors which determine where you find organisms, animals and plants. Temperature affects where you find animals. Some animals prefer, for example, warm temperature. Some prefer damp, cold areas. So that can determine availability of nutrients, which means food. Basically, if you have more food, you would tend to find more animals. This applies particularly to animals because plants can actually make their own food. So when we say nutrients, we're talking about food. Amount of light, mostly it affects plants, but some animals are also affected by light. So their animal distribution can be affected by light, but most of the time it's about plants. Availability of water and minerals. As you know, water is essential for animals, but water and minerals are really important for plants. So you would find more of certain plants when you have good availability of water. Availability of oxygen is important for both animals and plants. As you know, plants also respire, so they need it. But carbon dioxide is more important for plants. So that is a factor which can determine where plants are distributed, where you find more plants. It's important to recall at this point that photosynthesis is the process by which plants make food, their own food, but they need water mainly and carbon dioxide. With these 2 ingredients, very basic ingredients, water from the soil and carbon dioxide from the air, plants are able to make their own food using sunlight, energy from sunlight. And this is why these factors are really important for plants. Amount of light, water and carbon dioxide. Really, really important because photosynthesis depends on these factors. So let's get back to the original problem we started with. We looked at scientists exploring 4 different habitats of badgers, trying to find out and establish where the badger population is highest. So there are 4 different habitats. In each of these habitats, scientists make a footpath such as this, and they made sure that it's equal in length, and they used that as a transect. As you know, transect is a straight line. So if you imagine this as the wooded area, so the whole of this is the one wooded area, scientists had made footpaths like this which are equal in length, or they used footpaths like this, and that served as the transect. So that was they used this as the transect. As you know, transect is just a straight line. It can be anything. It can be like a line or a string, as long as it's nearly straight, that they can use it. So they sampled 4 such wooded area, 4 such habitats, but they used this footpaths as transects. Okay, so this is what they did. They walked along the footpaths and they counted the number of sets. As you know, sets are badger homes. This is where badgers live. In each set, an average of 7 badgers live. Okay, so let's assume that. So let's go back once again. So they're walking along this transect and look both sides of the footpath, and they count how many sets can be located in each of the habitat. Okay, so and then they counted each of them and recorded them. The more number of sets they find, they can estimate the population knowing the average number of badgers, or simply just knowing the number of sets that they have. They can say that if they find more sets in a particular habitat, they can say there are more badgers in that particular habitat. I hope you understand the scenario. Now let's go into the data. What is the control variable the scientists used in sampling the badger population? So have a moment to think about that. It's actually the length of the transects, or you can say the length of the footpath they used. Okay, so they have to make sure that these are the same in each of the habitat to be able to estimate, to be able to compare where they have more badgers living, because they're only looking for sets. Remember that. Okay, they're not actually counting the actual number of badgers. So that brings up a question in evaluation. We'll come back to the method they used later on in our evaluation. Let's look at the data. Here is what they collected. They collected the data and they made a graph using what they collected. Okay, as you can see, the description here is badger population in 4 different habitats. Okay, and data from 10 of each type of habitat. So they actually not sampled 4 woods. They went into 10 of each type of woods, as you can see, and this is the results here. Okay, so for each type of wood, they have sampled 10 such woods. Okay, and here you have the results. Now let's analyze this. This is how a typical exam question goes. You're given a graph and you're asked to analyze what it shows. Okay, so let's have a look at what this shows. The red lines that is shown here, what we're talking about is this red lines. That is the mean number of badger sets in each of the habitat. Okay, and this is the lowest number, and that's the highest number in that particular habitat, alders, as you can see here. Okay, so the first one is alders and deciduous, and then you have hedgerows, copses in here. Okay, so alders, deciduous, copses, and hedgerows. So you have these 4 habitats, and let's have a look at what the data shows us. Okay, now as you can see, straight away you can see which habitat has got more badgers. As you can see, you can straight away say that it's the deciduous one, because you have the mean number showing the highest of all of them, because if that is the mean number, you have on an average, that's where you have more badges, deciduous woods. Okay, but however, if you look at can you say that deciduous always has more number of badges than, for example, alders or copses? No, you can't, because in some woods you have more badges. For example, here, the highest number of badges sets found in this type of habitat, alders, is 8. In some habitats, it's in deciduous, it's low. So it's not, you can't say that for certain. Okay, so it's really important that you realize that what you see is a range of the number of sets in each of the habitat. Okay, so what is the range? Another type of question. Let's have a look at how else you can be asked about the data, asked to describe the data. Okay, so you can be asked like, what's the range of the badger sets in copses? Okay, so you can see here that the range of badger sets found in copses goes from 3, as you can see. If you go from left, from that point onwards, you go to 3. So you have from 3 to, at the top you have 10. So the range is actually 7. So 7 is the range of the number of sets that you can find in copses. You can now see the mean numbers on all the 4 different habitats that the scientists actually explored. You can see the mean number, the highest average you have is for deciduous woods, and you can also see the range of the number of sets that's in each of the habitats. And you can see deciduous has got the biggest range, the range being 16 take away 3, which is 13. But you can not say that the deciduous will always have a higher number of badges or badger sets than the hedgerows. The reason is this. Have a look at this. The lowest number you can find in deciduous is 3. So some habitats, deciduous type woods, have a lower number of badges than hedgerows, where in some places, in some types of or some habitats of this type, you have more number of badger sets. So the range is really important, and it's really important to realize that it's not always the case. Okay, so you should be able to analyze any data given and presented to you in the exam and be able to say the average number. Are you able to say that there is more living in a particular habitat using the mean values? Or you should be able to state the range, or be able to comment on comparisons such as between the habitats. For example, you may be asked to comment on where you least expect to find a badger. So you can say in this habitat, hedgerows, you can expect to find the least number of badges, as the mean number is the lowest in that habitat, and also the range is low. So generally you can expect to find less number of badges in this type of habitat. Pause the video now to read this additional information about badgers and where they live, badger sets. You will need this additional information to answer the following questions. 2 evaluation questions to consider in this particular example of estimating badger population or comparing badger population in different habitats. Can you explain how counting the number of badger sets give a scientist a more accurate estimate of the population of badgers than counting them individually? Okay, so how does this method of just looking for sets? Remember, scientists were looking for badger homes, the burrows and the holes on the ground, rather than counting badgers. The answer is this. Now, badgers move about, so it is very hard not to count a badger twice. So there is a possibility you count the same badger a few times as they move about. So it will not give you an indication of where you find more badges if you just count them. So that is a problem. That is overcome by just counting how many badger homes are there. This gives a better idea about where you have more badgers living. So that is one very important advantage of counting something else instead of the organisms themselves, which gives you an idea about the number of organisms living in a place. As you can see, badgers move about, and it is very hard to not to count badgers twice or even 3 times. Now the second question: explain how this method may not give you an accurate idea of the badger population. Okay, so here you looked at an advantage. This is asking you to give a disadvantage of this method. And here is the answer: badger may no longer live in some of the sets. So you're counting the sets, but there's no guarantee badgers actually live there. They might have moved away from the badgers, or maybe even died off. So badgers might have died, but you will be still counting the sets. So that can give you an inaccurate picture of how many badgers actually live in a habitat. There are more reasons for why this method may not give you an accurate idea about where you have higher badger population, and these are here. Badgers might have died. Sets may be occupied by other animals. So if you read the passage that came before this question, you would have realized that these sets, which are badger homes, are occupied by other animals as well. So by just counting the number of sets, you may end up getting the wrong idea about the number of badgers that live there. In this example, you have an area, part of the park maybe, and you have a tree here and a patch of green containing made of grass, as well as dandelions. So each of these green streaks are grass plants, and you have these yellow blobs represent dandelions. Okay, so you have this scenario. You also have a footpath where people walk and trample all the anything on that footpath. Okay, so that is the scenario. You have to come up with a method to measure the distribution of the grass as well as the dandelion plants from the foot of the tree as you move out towards the path. So the distribution that way, how you would figure out scientifically using a transect, which is a tape measure, or even a string with regular markings, regular intervals, and a quadrat, which is a square frame, usually 1 meter square in area. Okay, so that is a question. Can you describe a method? Here is the method. You put the transect, the belt transect, along a length at random, and you already have markings on it. So the transect is laid straight from the foot of the tree right across where you want to sample. The next stage would be to sample or place the quadrat at regular intervals. It's already been marked, the tape measure has been marked, regular intervals, so you just have to place the quadrat at those intervals and measure the number of each species of interest. So in this case you would measure or count the number of grass plants inside the quadrat, inside the square, and you'll also count the number of dandelions inside the quadrat. Now you should also plan to collect valid data, which means it should be along the transect, equal distance from each other, so the evenly spaced markings. You will place the quadrat along those markings, or on those markings along the length of the transect. Okay, you should also mention or pay attention to how you will count the dandelion plants if it is on the edge of the quadrat. Say for example, if your quadrat is like so, that, as you can see, that dandelion is just on the edge of the quadrant. So how would you deal with that? Now, if it is more than half of the plant inside the quadrant, you would include in your counting. So you should mention that in your method. You should also plan to use more transects or repeat of the same procedure. So you would repeat the same thing over across different lengths of the same area. So you're interested in the distribution of this area, so you would repeat it several times, and that improves the validity of the data that you collect. Here is the summary of the method to scientifically measure the distribution of species using a transect, so and some quadrats. Make sure you read this and understand the points. Let's have a look at the data obtained by the method described just now. So here you have the same diagram, tree and the plants, grass as well as the dandelions. Okay, so but you can see as you move away closer to the tree, you have less grass and dandelions. A few only here near closer to the tree. As you move away, you can see there is a thicker population of these two species, grass as well as dandelions. And as you move away, you have less and less of grass and dandelions, and then again grass. There is more here. So that's what we call as distribution. But if you measure this scientifically using the method just described, using a transect and a quadrant, you get proper data. And different types of charts can be drawn with the data obtained. But here is an example of such data. Many questions can be asked from a chart like this, and this type of chart is known as a kite chart. You don't need to know the name of the type of chart, but just out of interest. Okay, so you have the chart here. What does that show? You can see how the number of these two different species change as you move away from the tree. So here is where the tree is 0, represents the position of the tree. And for example, look at the dandelions. You can see the maximum number is found around about 5 meters away from the tree. And it's the same with the case with the grass. You have more of them as around about the same distance away from the tree. And the numbers go down nearly, it's very small amount present here, very small amount present here. Now, additional information will be given in the exam. For example, this can be marked as the footpath. Okay, so, and then you should just be able to explain what's happening, with regards to the numbers. Why the numbers are very small, close to the tree. Easy. Plants compete for sunlight, and the tree being big, it blocks most of the sunlight reaching the grass as well as the dandelions. It looks like grass survives better under the tree, as the plant is small. The grass is really small plants, so it can make use of diffused sunlight perhaps, whereas dandelions need a little bit more sunlight, so you don't have any under the tree, and the numbers gradually increase. And when it's 5 meters away from the tree, it's getting good sunlight, and it's no longer blocked by the tree. That's one very good reason to give if you're asked about why, or can you just explain the distribution. If you're asked, then you can give that reason. Okay, light being one reason. Is there any other reason for this kind of distribution? Water. Plants compete for water and also minerals from the soil. So as you can see, dandelions cannot compete with the tree being massive and it's got a good root system to absorb water and minerals from the soil. You don't get that. So dandelions lose out in the competition, and you don't find any closer to the tree. So many ways of explaining, but you need to use valid points only. You can't just list the factors that we saw at the start of the video. You need to pick the valid factors which are applicable in each of these situations. Okay, same with the grass as well. It's not able to compete much, a lot with the tree, but it's growing in good numbers closer to the tree, possibly because it requires very less water and it can absorb the soil water which is on the top surface of the soil, and that is possibly enough for some of them to survive. You'll also notice closer to the footpath here, you'll see less of these plants surviving, possibly because people walk all over the plants, and the plants die in the footpath. So not many survive where the footpath is. And as you move away from the footpath, you can see more of the grass plants, whereas the dandelions are nearly going to zero. So that is a distribution. You may be asked, where is the maximum competition between the grass as well as the grass and the dandelion plants? You can see that's where, because this is exactly where maximum competition between the two species takes place. So you should say at 5 meters the competition between the species is maximum. Let's have a look at this example where a scientist is actually looking for the percentage cover of algae on rocks. Okay, so how much of the rock is covered by, what percentage of the rock is covered by an algae which is growing on top of the rocks. As you can see, as you can imagine, you cannot count the number of algae because as it's very small, so you would require to calculate percentage cover, which is done this way using a quadrant technique, but instead of counting, you look at the area of the algae that is growing inside the quadrant, inside the squares inside the quadrants. Let's have a look at that. That is that this diagram shows the algae growing on rock, and where you place the quadrant, some parts are covered by algae, as you can see here, here, here and here, but there is no algae growing here. So what percentage of algae this represents? Obviously you have to repeat this several times, but this only shows one quadrant. So from this quadrant, can you say what percentage of algae is growing on the rock? So we need to estimate the number of, or the area that is growing. So if you look at the total area of the quadrant, let's just say 1, 2, 3, 4, 5, 6, 7, 8, and 4 times 4 is 16. So you have 16 squares here. Okay, so the total number of squares you have is 16. Out of that, you need to figure out how many are covered by the algae. So you need to count it a bit carefully. It's a bit tricky. So you can see here, this is half. Count this as half. That as a quarter. You may want to count this 3 as 1, and that is approximately half. And that is a half, and that's definitely a half. Okay, so you may not count this at all, and so use all of this, approximations to estimate the number of squares it actually is covered. To make it slightly easier, you may want to number it. Okay, here is something with numbers. It should be easier now, and as you can see, this is counted as 1, 1, 2, 3, 4. Although there is a little bit less here, of area not covered, you can almost see it's nearly covered. So 1, 2, 3, 4, 5. Count that as half, that's 6, and 7, 8, and as you include this in that and that in that, so that's half. So totally you have 5, 6, 7, 8, and 8.5. So that's the coverage of algae you have on the rock inside this particular quadrant. So the percentage coverage of the algae can be calculated using usual percentage calculation. So that's the covered area where the algae is growing. That's the total area of the quadrant. So we know it's 16 squares is the total. So we write the covered area we just worked out is 8.5, 8.5, 16. So this works out to 53% of the total area of the rock covered by the algae. How would you improve the accuracy of this estimate? Obviously, you need to take more samples. The more sample you take, the accuracy of estimate improves, so you get more accurate estimate of the percentage of cover. Here is another example showing barnacle distribution along a shoreline. Okay, barnacles are small creatures with shells, normally clings on to rocks along the seashore. Okay, so you have four different types of organisms which live close to the shores. You can see how they are distributed as you move away from the shoreline. So that is the seashore where the water meets the land. And as you move away, you can see the numbers increase for some of them, or nearly all of them it increases. So if you look at the abundance of these species as you move away from the water, you can see at 3 meters you have more rock periwinkle here. So you should be able to describe that in terms of the numbers as well as you give possible reasons. So they may need damp conditions for them to survive, or their food may be in from water. So as the water moves in, the tides, with the tide, so they may get food from the water. So the source of food may be in the water. Okay, so that's why they need to be closer to the water line. So you have rock periwinkle really close to the water. As you move further away, you have increasing population of acorn barnacles around 5 meters from the shoreline. You have then competition between these three species. As you can see, limpet population is increasing here. In fact, that's the maximum in that place. So the competition is with these three species at 5 meters. That's worth mentioning if you're asked to describe it. Okay, so which one prefers the driest conditions? So which one lives furthest away from the shoreline? It's this one, knobbed periwinkle. As you can see, it's expanding out here, getting slightly bigger. As you can see, as you move away from the shoreline, the knobbed periwinkle increases in its population. As you can see here, it's just about increasing, so its maximum is just around here somewhere. Okay. This is the last example where you have strawberry plants, spelt wrong here, but it's strawberries. So you have two sets of data collected from two different fields. One is a sunny field and the other one is shaded. So this does not get as much sunlight as these plants here. So the farmers wanted to know, obviously, where it produces more strawberries. So as you can see, this is a different type of graph. You need to analyze this a bit more carefully here. The number of strawberries is on the x-axis. So for example, this plant here produces 10 strawberries, and the number of plants which produces 10 strawberries is around about like 2 or 3, as you can extrapolate onto the left here. And if you look at the number of strawberries producing 15 strawberries, that's around 30 plants. I hope that gives you an idea about how to analyze this. If you look at the difference between them, you can see there are lower number of strawberries produced generally. The maximum you can see is about 13. Here you have 17 strawberries produced as a maximum. There are 5 plants, around 6 plants, which produce 17 strawberries, and you have the highest number limited to 13. So this clearly shows the plants which are in the sunny field produce a higher number of strawberries generally, and the strawberry plants in the shaded field produce less. The reason? It's very obvious. They get more sunlight. More sunlight means more photosynthesis. More photosynthesis means they're able to produce more glucose, which means more strawberry plants. You know, the glucose is stored in plants and in fruits, seeds, and in their roots. So more sunlight, more strawberry plants, and that's what this data shows. The other type of questions that you may get is what is the mode of the number of strawberries in the sunny field? Okay, mode, as you know, is the most occurring number. Okay. So in the case of the sunny field, the mode would be 15. As you can see, there are 30 plants with that number. So the most often repeated number or value is your mode.
So 15 would be the mode for the strawberry plants in the sunny field. So what that means is, most of the time you will find 15 strawberries in the strawberry plants living in that are in that area, in that sunny field. Whereas the mode for the strawberry plants which are in the shaded field would be 6. As you can see, that bar is the tallest one, so that means there are around 32 plants which only give about 8 strawberries. So 8 would be the mode here. For the strawberry plants in the shaded area, the mode would be 15. So that is another indication of that you do get more strawberries in the sunny field compared to the shaded field. So sunny field more, shaded field less. What would be the control variable in this case? Now they have sampled 100 plants. So if you added all these numbers together, you should get 100, and that is 100 strawberry plants have been sampled. Another control variable which is not very straightforward is that they should actually sample at the same time of from the time of planting these strawberry plants to when they actually do this do this investigation. So the time for them to grow and produce these strawberry plants should be the same for each field. In this video, we have only just looked at some examples of how you should describe distribution of organisms and how you would sample them and your use of transects and quadrats. So we have looked at how you would use a transect, what is it in the first place, how you would use it, and how you would use a combination of transect and quadrats to find the distribution of different species along the length of a habitat or across a habitat. We also looked at some evaluation of methods, what makes the method good and what could possibly go wrong. Now, in fact, in exams you will be presented with loads of different varieties of data, but we have to employ similar techniques. We should be able to analyze and describe, giving the usual reasons that explains the distribution. Okay, so we also looked at mean, median, and so on. So you should be familiar with these terms, mean, median, and range, and be able to use it to analyze data. And you'd be glad to know that is the end of this a little bit longer lesson. Apologies for the length of the lesson, but you really need to know all these techniques and be able to analyze data in those using the techniques that we've described in this video. Bye for now.